So in last class we understood about trigonometric functions sin theta, cos theta, tan theta, cot theta, sec theta and cosec theta. Okay. And we understood that if we have to find out angle value like 120 degree, how to find? We can break 120 degree into two parts, 60 plus 60. We can use here sin 2 theta formula. Otherwise, we can use a plus b formula also, like we can write 120 90 plus 30. Okay. So other formula also we discuss for sin as well as cos theta formula, cos a plus b and tan a plus b. These formula we discuss, and simultaneously we discuss 2 theta formula for all three functions, sin 2 theta, cos 2 theta and tan 2 theta. For cos 2 theta we discuss three formulas, right? Cos 2 theta we discuss three formulas: cos square theta minus sin square theta first, second 2 cos square theta minus 1, and third 1 minus 2 sin square theta. Okay, these we discuss. Now understand here this point also. Actually 2 theta formula you all know, right? 2 theta formula, sin 2 theta formula you know. How much? 2 sin theta cos theta. Okay, but here one point extra. What? Suppose only sin theta is given. Can we write sin theta formula? Yes, we can write. How? Understand. If here 2 theta, then here theta, theta. Agree or not? Half angle it is. This is 2 theta, then it is half theta. This is also half theta, right? Now, if sin theta formula we have to write, then we have to break into half angle. So this sin theta formula will be 2 sin theta by 2 cos theta by 2. Okay. And remember this point, these all formulas we will use in physics. In many chapters we use all these formulas, right? Okay. So sin theta formula we can write 2 sin theta by 2 cos theta by 2 half angle. Here it is 2 theta to half angle. Theta to half angle, theta by 2, theta by 2. Similar like that, if suppose cos 2 theta formula we know it is how much? 1 minus 2 sin square theta. Otherwise cos also we know. You can write all these remaining formulas. Okay. So suppose if cos theta is there, to cos theta how we will break? So here 2 theta, then half angle theta. So if here theta, then half angle is theta by 2. Agree or not? So it will be 1 minus 2 sin square theta by 2. Is it clear? Understood? Cos theta, then it is 1 minus 2 sin square theta by 2. Otherwise we can write it 2 cos square theta by 2 minus 1. Clear? Okay. So yes, we understood half angle formula also. These are important. Sin theta and cos theta, these are important. Is it clear? Okay. Now we will understand today graph of trigonometric functions. Okay, this is important topic. Graph of trigonometric functions. This is very, very important. Now all of you listen it very carefully, understand it. So first we will discuss sine graph, sine function graph, sine theta graph. How many of you know sine theta graph? So first point, this is very important, है ना? Sine theta graph, sine values you know na? Sine 0, 0. Sine 30, 1 by 2 means 0.5. Then sine 45, 1 by root 2 increases. Sine 60, root 3 by 2 again increases. Sine 90, 1. So this is maximum value of sine theta. Sine function maximum value is 1, where it is at 90 degree. So if we talk about 0 to 90, then continuously sine function value is increases 0 0 but 91 maximum. Okay, so sine function sine theta values increases as theta increases, but it is till 90 degree, because at 90 degree it is maximum. So you know, if any quantity reaches at maximum level, then after it will decrease. So sine theta graph is like this. You have to remember, very important. Okay, I am drawing it. So sine theta graph is like this. Okay, now angle also I am mentioning. This angle is 90 degree, and 90 degree value is how much? 1. This is 1. At y-axis we taken sine theta, and at x-axis we taken theta. Clear? Okay. Now very interesting point is what here, that 0 to 90 values are increases, right? Graph rises means values increases. Okay. After 90 values decreases, and here this sine theta value becomes 0. You know, at x-axis y values are 0. This is how much angle? This angle is 180 degree. This angle is 180 degree. Okay. This angle is how much? This angle is 270 degree, and interesting point, at 270 degree value is -1. Value is -1, where at 270 degree, so this angle is 270 degree. Again at 360 degree, here values is 0. Okay, so again at 360 degree, this is 360 degree, value of sine theta is 0. So interesting point is what in this graph? Understand, first increases, then decreases, then negative values. Okay, in negative it increases, and again it becomes 0. Now very important is what you can see, this graph shape is like this. Okay, after 360 degree, again its shape is repeating itself, right or not? First like this, same shape repeated after 360 degree. Increases, okay, then decreases, yes, decreases, negative, yes, negative, increases in negative, yes, right or not? So sine theta graph, this sine theta graph, what it tells us? It tells us that this sine function repeat its values or repeat its structure after a certain angle. That certain angle is how much? 360 degree. After this angle, again it repeat itself. Such functions are known as periodic functions. Understood? Periodic function. Rohan, understood? Rohan, periodic function. Periodic function means what? After a certain value of angle, a function repeat itself. Such functions are known as periodic motion. And in NEET, they will ask this question. How they will ask in physics? Which function represent periodic motion? Because in physics we discuss about motion. So they will ask which function represent periodic motion. So this sine theta function represent periodic motion. How? Because we can see after this angle 360 degree, it repeat itself. That's why it is periodic function. To write this point, sine function is periodic. It repeat itself after 360 degree. Understood? Okay. Now here important point is what? You can see this function has positive value as well as negative value. Agree or not? And you can see these positive value and these negative values both are equal. Positive value, negative value both are equal. 0 to 180 degree positive, 180 to 360 degree negative. Positive values and negative values both are equal. So sometimes they ask what is the value of sine theta for one cycle, or in other words they can ask find the average value of sine theta. What? Average value of sine theta. Average is calculated for one cycle, half cycle, they can ask for any cycle. So generally they will ask for complete cycle. To this point also you have to remember, for complete cycle average value of sine theta is 0. Understood? Okay, because positive values, these positive value and negative value both are equal, that's why we can say that average value is 0 for one cycle. Next point is 180 to 360 values are negative and 0 to 180 values are positive. So we will understand one more point, that when sine theta value will be negative, when it will be positive. So by this graph we can say that 180 to 360 it is negative and 0 to 180 it is positive. Now we will understand cos theta graph. To cos function graph, both are very important, sine theta and cos theta. This cos function graph is like this. You know, sine function graph start from 0, but this cos function graph does not start from 0. This is the difference. And you know this point also, sine 0 is 0, sine 90 1, but cos 0 is 1, reverse values. Cos 0 1 and cos 90 0, reverse. Agree or not? So in case of cos function, graph will start from maximum value, okay, 1, then it decreases and like this. Okay, we can extend. Sometimes we call them cos wave, sine wave, because like wave it is continuously travel, like wave it continuously travel, that's why we call them cos wave or sine wave. Okay, now understand here, this is maximum value 1, and where it is 1? At 0 degree. Then at 90 degree it is 0. This point you have to remember, 90 degree it is 0. Then 180 degree, this is 180 degree value. At 180 degree it is minus 1. This is 180 degree and here it is minus 1. Okay. Then this is 270 degree. At 270 degree again it is 0. And again it is 1, where at 360 degree here it is 1. Clear? Okay. So important point is what? Sin theta become maximum at 90, but cos theta become 0 at 90. Cos value is maximum initially. Okay, so 0 to 90 it decreases. 0 to 180 we can say. 0 to 180 continuously it decreases. Then 180 to 360 continuously it increases. 0 to 90 values are positive. 90 to 270 values are negative. Then 270 to 360 values positive. Now who will tell what is the value of its time period, or we can say after which angle it start to repeat itself? Send your response. I am waiting. Here we discuss in sin theta that it repeat itself after 360 degree. What about cos theta? cos theta start to repeat itself after how much angle? Send your answer. Cos theta start to repeat itself after how much angle? In sin theta we discussed that it start to repeat itself after 360. What about cos theta? Some students are saying 270, some are saying 360, 270. Most of the students are saying 270. No, 270 is wrong. 360 is correct. Open your eyes, what you are doing. What I said, that after how much angle it start to repeat itself. Okay, so first repeat means whatever initial shape, that again it will come after how much angle. So it is like this: initially decreases, different shape, different shape, different. You can see here after 360 its shape is same, shape is same as that initial, right or not? Here decreases, here also we can say after 360, after 360 decreases, right or not? So its time period also 360, right? We can say that it repeat itself after 360 degree. So both are similar. Okay, now next question for you. Next question for you. Here we discussed that for complete cycle average value of sin theta 0. Now what is the average value of cos theta for complete one cycle? Who will tell answer? Send your response. I am waiting. What is the value of average value for one cycle? Some students are sending one continuously, one, all are sending one. I don't think that how all can do mistake. Actually, yesterday most of you given correct answers every time, but today you are missing the line. Some students are sending only correct. Why? Because today these points are more conceptual. So today is your test. Those who are logical, those who think, they can give correct. Now they are sending 0. Yes, very good. What we have to find out? Average value for one cycle. Here you know one cycle completed, right? Now see it. This part positive, this part positive, but this part negative, this part negative. And one more point, that this part, only this part and this part both are equal or not. So this is positive, this is negative. If we add them, positive and negative values, it becomes 0. Again this part negative, this part positive. If we add them, only this part, negative plus positive, again it also becomes 0. So very interesting that average value of cos theta for one cycle is 0, and for sin theta also average value 0. So what is the similarity between both function? Both repeat them after 360 degree and their average value for one cycle is 0. Okay, so here also we can say that cos function repeat itself after 360 degree and its average value is 0 for one cycle. Understood? Okay. So we understood both graphs, sin theta and cos theta. And one more point here we have to observe, that if we take any particular angle, any particular value, suppose this value, right? Suppose this value. So here we taken this value. So we can say this value is possible for two angle. One angle is this one and another angle is this one, right or not? A particular value is possible for two different angle. One angle is this one, another angle is this one. Means one is between 0 to 90 and next angle is between 90 to 180. Right or not? This is also interesting. What? That in sin theta graph, a particular value is possible for two different angle. One angle is between 0 to 90, another angle is between 90 to 180. Right? Only one graph, how many things we are observing? Same here. Here also, any particular value. Suppose we taken this value. So this value is possible for two different angle. You can see here one angle is here and next angle is here. Right or not? So one angle is between 0 to 90 and next angle is between 270 to 360. Is it clear? Okay. Both function are periodic. Sin theta, cos theta, both are periodic function. This point also you have to remember. Both sin theta and cos theta are periodic function. Periodic function already we understood, those functions which repeat themselves after a certain angle, certain time we can say. Okay. So both sin theta and cos theta are periodic function. We understood. Now next. Now we will understand how to find value of sin 270 degree, sin 300 degree. Yesterday we discussed that, okay, 120 we can calculate, 150 we can calculate. We can break them into two smaller angle, those we know. But today we will understand how to find values of angle which is greater than 180, like 300 degree. Okay. How to find 210 degree, 270 degree, how to calculate, understand. So first we have to understand quadrant system. Okay. So this completed this one. Now next is quadrant system. So write quadrant. So actually we break 360 degree into four quadrant. One is 0 to 90 degree. Okay, 0 to 90. Second is 90 to 180. And next is 180 to 270. And next is 270 to 360. Clear? What we do? We break 360 degree into four part, 0 to 90, 90, 180, 180, 270, 270, 360. Why we break? We can understand by this graph. You know, everywhere you have to think why we are doing. By this graph you can understand. This graph also breaking itself or changing after 90 degree, 0 to 90, 90 to 180, 180 to 270, 270 to 360, right or not? So we break here this 360 degree into four quadrant. Okay. Now in first quadrant means 0 to 90. All functions are positive. All functions are positive. All function means sin theta, If sin theta positive, then you know cosec theta already positive because sin theta equal to 1 by cosec theta. Okay, cos theta and sec theta. Same here also, cos theta positive, then sec theta also positive. Tan theta and cot theta. Now you can say, sir, why these are positive? So actually in first class in yesterday we discussed this point that all these trigonometric functions we define for right angle triangle. So do one thing here, draw right angle triangle. Okay, so here we drawn this right angle triangle. Okay, you know this side is, if Cartesian system we understand, then this side is positive, this side is negative, this side is positive, and this side is negative. Agree or not? Plus x minus x plus y minus y, right? So here this is perpendicular, this is base, and this is hypotenuse. Okay, suppose this point is O, this point P, and this point Q. Okay, so its baseline OP in this direction, so it is positive, this direction is positive, so OP is positive here. Okay, OP is positive, and PQ, P2Q, PQ also positive. We can say that OP is what? Base is positive, and PQ is what? Perpendicular also positive. Now any trigonometric function, sin theta, cos theta, whatever, in those this base and perpendicular will come. Hypotenuse is distance, hypotenuse always positive. OQ, hypotenuse always positive. It is always positive because it is what? Distance. Right? Okay. So sin theta, if we write sin theta, so sin theta is what? Perpendicular divided by hypotenuse. So perpendicular positive, so sin theta positive. Cos theta base by hypotenuse, again base positive, so cos theta positive. Tan theta perpendicular by base, both are positive, so tan theta also positive. That's why all these trigonometric functions have positive value in first quadrant. Is it clear? This angle is theta. Now come back in second quadrant. So this is second quadrant. Here this is what? Right angle triangle. Now what is positive here? Tell me, its base is positive or negative? So here this is suppose S and this is T. So tell me, base is positive or not? Send your response. Base is positive or not? Base is positive or not? Send your response. No. Yes, very good. So it is not positive. Base is negative here because this direction is negative. So base is negative. So this time base is negative. Okay. Now what about perpendicular? Perpendicular ST. So perpendicular is positive or negative? Perpendicular positive or negative? So perpendicular like this, and this direction positive. So perpendicular is positive. Clear? Okay, so we understood perpendicular positive and base negative. Hypotenuse always positive. Now sin theta, check sin theta positive or not. So sin theta formula, perpendicular by hypotenuse. perpendicular positive, hypotenuse always positive. So sin theta is positive in second quadrant. Now sin theta positive, so cosec theta positive. Cos theta. Cos theta is base by hypotenuse and base is negative. If base negative, तो cos theta also negative. Cos theta negative, तो sec theta negative. So cos values are negative in second quadrant, means 90 to 180. Then check tan theta. Tan theta is perpendicular by base, perpendicular positive but base negative. Perpendicular positive but base negative. So it is perpendicular by base, it will be negative because perpendicular positive but base negative, so overall finally it will be negative. So very important, in second quadrant only we can say that only sin theta and cosec theta are positive. Only sin theta and cosec theta are positive. Other are negative. These are positive. Other functions are negative. Okay, so in second quadrant, in first quadrant all are positive. Okay, we understood already. So here finally I am writing the summary. In first quadrant all are positive. In second quadrant sin theta and cosec theta positive, remaining negative. Remaining other negative. Now in third quadrant, can you tell which are positive in third quadrant? Can you tell which are positive in third quadrant? Send your response, I am waiting. Which are positive in third quadrant? Send your response. Send your response, which are positive in third quadrant. I am waiting. Okay, very good, very good. Most of the students are sending their answer. Tan theta and cot theta are positive. Very good. Now I can say that you are listening and you are understanding the concept. Very good. I am very happy. So in third quadrant, this triangle will be like this. Okay. Now here this triangle is like this. Here we can see that base is negative and this perpendicular also negative. So in third quadrant base is negative and perpendicular also negative. Both are negative. Now check which are positive. Sin theta formula perpendicular by hypotenuse. So perpendicular negative. So sin theta negative. Cos theta base by hypotenuse, base is negative, to cos theta also negative. So both are negative. Now tan theta. Tan theta is, if we check here, so tan theta is perpendicular by base, to perpendicular negative and base also negative. Negative negative cancel out. Finally we will get positive value. Right or not? So we can say in third quadrant only tan theta and cot theta are positive, remaining are negative. Now check in fourth quadrant. Send your response in fourth quadrant which is positive. Send your response. Okay, very good, very good. Most of the students, they are sending cos theta and sec theta. All are very good. How it is possible that all of you are giving correct response? Very good. Cos theta and sec theta are positive, other are negative. Means you understood this concept. So finally in this fourth quadrant, if we draw this right angle triangle, so we can say that this time base is positive and this perpendicular is negative. So base is positive and perpendicular is negative because it is in downward direction and it is negative. Base in this direction, it is positive. Okay. So base positive, perpendicular negative. Now wherever base will come, those are positive. Perpendicular will come, those are negative. So base come only in cos theta and sec theta. That's why they are positive. Now you can say, sir, in tan theta also base come, but in tan theta perpendicular also come na. Perpendicular by base, negative by positive, finally negative. Right or not? So here cos theta is positive, cos theta sec theta positive, other are negative. So in fourth quadrant, in fourth quadrant, cos theta and sec theta are positive, other are negative. Other are negative. Okay, so we understood about these trigonometric functions, that where these are positive, in which quadrant these are positive, in which quadrant these are negative. Okay, now how we will use these all concepts, that these are positive, negative, okay, we understood, but where we will use them? Understand this point. Understand this point. Actually, suppose if they ask that find sine 300 degree, so how we will calculate sine 300 degree? Or any function, not only sine, tan 300, cos 300, how we will calculate it? Understand. So what you have to do, you have to write this 300 degree in terms of these mention angle. First check 300 degree is in which quadrant? So 270, 360. So 300 is between 270 to 360, means 300 is in fourth quadrant. Now write 300 degree in form of 360, otherwise 270. Clear? Write 360 degree in form, write 300 degree in form of 360, otherwise 270. How? Understand. Sine 300 degree we have to find out. 300 is in fourth quadrant. Now fourth quadrant means 270 to 360. So what we are doing, we are writing this 300 by using 360. Okay, so we can write 300 degrees sine 360 minus 60. Agree or not? Sine 360 minus 60. 360 here minus 60. So if we subtract, then again we will get 300. Yes, we can write. Okay. Now sine theta is in which quadrant? Fourth quadrant. In fourth quadrant, sine theta is positive or negative? Check. So yes, we know sine theta is negative. Only cos theta and sec theta are positive, other are negative. So this 360 degree meaning is only one. What? That here this sine theta is negative. So directly what we can write? Remove this 360, only just what will remaining here? 60 degree. 360 minus 60 means fourth quadrant, and fourth quadrant means sine theta negative. So we can replace this 360 by minus sign. and finally we can write it sine 60 degree. Is it clear? Understood? So sine 300 degree is equal to minus sine 60, and you know sine 60 value is root 3 by 2. So sine 300 degree is negative, and how much it is? It is minus root 3 by 2. Understood or not? One more example. Suppose you have to write sine 300, we understood. Suppose you have to find out cos 300. Okay, so what we will do? First we will check where it is. So it is in fourth quadrant. Okay. Now we can write 300 by using 360. Cos 300 equal to cos 360 minus 60. Yes. Now check this cos theta is positive or negative in this quadrant. So cos theta is positive. Okay, in fourth quadrant. So 360 meaning is what? That here this cos theta is positive. Okay, and finally angle 60. So cos 60. Cos 60 value 1 by 2. So cos 300 value is equal to plus 1 by 2. And we can check these values by graph also. Before that I explained this concept. You can see here 0 to 90 degree values are equal to 270 to 360. Right or not? To cos 60 1 by 2 and cos 300 also 1 by 2. Is it clear? Okay. So we understood how to find angles which are greater than 90 degree. So two example only we discussed today. Remaining we will discuss tomorrow. So tomorrow I will explain you other like 10, 300, 10, 270. How to find all those angles tomorrow we will understand. Okay, so whatever we discussed today, revise all these points. Okay friends, so this class over now chemistry class.
Yesterday we did a degree of carbons, right? First of all we started with organic chemistry definition, then we started classification of organic compounds, and then we did degree of carbon, right? And you did very well in those topics. Okay bacha? Now today we are doing discussions about degree of alcohol, degree of halide and degree of amines. Okay bacha? Good morning everyone. Very good morning. So now next point I am writing here, that is degree of alcohol and halide, right? Degree of alcohol beta and halide. X means what? Halogen, right? X means halogens like fluorine, chlorine, bromine, iodine. You will understand this. Okay bacha? So in this you will see, first of all how we decide. I am writing here degree of alcohol and halide. Degree of alcohol and halide will depend on degree of carbon. Will depend on degree of carbon having OH or halide, right? So you will check the degree of carbon on which OH is present or on which halogen is present, and that will tell you the degree of alcohol and degree of halide, right bacha? I am giving simple example here. Look at this. Imagine we are having this example. I am saying here it is CH3, it is CH2, and here it is having bacha, it is having OH. Imagine this one. Now here we have CH3, CH3, CH here, and here we have OH, right? And here we have CH3, C, CH3, CH3, okay, and here we have OH. Fine. I have given you 3 examples of alcohol, right? So which alcohol is primary alcohol, which alcohol is secondary alcohol, and which alcohol is tertiary alcohol? Fine. If we check, tell me the degree of carbon on which our alcohol is attached. Our OH is attached, bache. You see carefully. This carbon is a primary carbon because it is connected to only one carbon, right? This carbon which is having OH is secondary carbon. That's why I can call this alcohol as what? Secondary alcohol. And this carbon on which our OH is present is a tertiary alcohol, right? It's a tertiary carbon. So I can say from here that this is a primary alcohol. This is a primary alcohol, right? This is what? This is a secondary alcohol, and this is a tertiary alcohol, right? So very, very easy to understand. Clear, bacha? Very easy. Just check the degree of carbon, and from that you can tell your alcohol is primary, secondary or tertiary. Same way you can go for halides also. Same way, beta, you can go for halides. Please answer this question again. Here we have, I'm taking example. We have here CH3, CH2, CH2, and this is having halogen. It is having halogen. X means halogen, okay? Now, next thing I am saying, we are having here CH3, C, CH2, CH3, imagine this way. And here we have halogen. And next, which I am saying here, we are having CH3, C, CH3 here, CH2 here, assume CH3 here, and this is our halogen. So please tell me, what is this? First, second, third. Tell me degree of halides. Degree of halide depend on degree of carbon. Yes, everyone, please make it fast. Fast. Decide the degree of halide or halogen based on degree of carbon. Monica is saying right, primary, secondary, tertiary, primary, right. Very good. Very good. You see, bacha, this carbon which is having halogen is a primary carbon. That's why our halide will also be known as what? Primary halide. Primary halide. Very good. The carbon having halogen is a secondary carbon, right? That's why our halide also which one? Secondary halide. Our halide also secondary halide. Very good, right? Next one, the carbon with halogen. The carbon with halogen is a tertiary carbon. This is tertiary carbon. That's why we can say this halide is a tertiary halide. This halide is a tertiary halide, right? I'm saying once again, the degree of alcohol or degree of halide will depend on degree of carbon on which our OH group or our X group is present. Clear, bacha? Very easy. Okay, very easy. Nice. Now, next thing is degree of amine. This is a little different. This is a little different, so we are coming to degree of amines. Next, bacha. The next one is degree of amines. Right? Degree of amines. Clear, bacha? So degree of amines, how will decide? It depends on number of carbon attached to nitrogen atom of amine, to the nitrogen atom of amine, right, beta? It is not like the previous cases. It is little different. It is depending on the nitrogen. To that nitrogen, how many carbons are attached, that will decide the degree of amine. Clear, bacha? If we take example here, look at this carefully. We are having here CH3, CH2, CH2, and imagine here we have NH2. Clear? So our nitrogen is attached. Our nitrogen is attached to what? How many carbons? Only one carbon, right? Similarly, beta, if you check carefully, CH3, CH2, and here we have nitrogen. Here we have one more carbon like CH3, and here we have hydrogen. This nitrogen is attached to how many carbons? 2 carbon, 1 and 2. Clear? And similarly, if I go beta for third one, it is what? It is our CH3, CH2, and here we have CH3, and here we have also what? CH3. Clear? If we check this case, here our nitrogen, first tell me, this amine is primary amine, secondary amine, tertiary amine. Yes everyone? First one. This first one is primary amine, secondary amine, or tertiary amine? Answer me fast. This is a primary amine, secondary or tertiary? Yes everyone? Very good, very good. Why? Because nitrogen connected to only one carbon. Yes, very good. It's a primary amine. It's a primary amine. Very good. Right, bacha? Primary amine. Next one, if I say this nitrogen is attached to how many carbons? 1 carbon, 2 carbon. So this one is which one? Primary amine, secondary amine? This nitrogen is attached with this carbon as well as this carbon. So it is primary or secondary? Very good. It is a secondary amine. Very right. Clear, bacha? Next one, this nitrogen is attached to how many carbons? 1 carbon, 2nd carbon, 3rd carbon, right? That's why this amine is what? This amine is a tertiary amine. This amine is a tertiary amine. I hope you clear. I hope you are clear here. Fine. The number of carbon attached with nitrogen will decide the degree of amine. Okay. It is not like the other ones. Okay. So please give this answer. You have to find degree of amine in given options. Right. First one is, here we have CC, NS2. Second one is CC, NS2. Third one is C, NS2. And fourth bit I am saying, imagine we have CC here, C here, NS2. Please tell me. Give me sequence wise. Means it is primary amine, secondary, tertiary. Just give me this way, sequence wise. Tell me the answer very fast. Decide degree of amine. First one is primary, right. First one is primary, very good. First amine is primary. Very good. Navya, Truthi Naya, give the right answer. Very good. Very good. Look at this beta. Nitrogen is attached to only how many carbon? 1 carbon. It is a primary amine. Next one, this nitrogen is attached to again only 1 carbon. It is also which amine? Primary amine. Right? Again here this nitrogen attached to only 1 carbon. It is also primary amine. Clear? And here also beta attached to what? 1 only, so it is primary amine. All are only primary amines. All are what? Primary amines. Very good. All are primary. Very good. Fine. Now, see the next question. I hope we are clear here. Degree of amine depends on number of carbon attached to nitrogen. Right? Be clear about this. All are primary. Now, next thing. Question is degree of alcohol I am asking. So please give answer carefully. First one is, here we have CC, COH. Second is C beta OH. Third is CCC OH. And fourth one is C here, carbon, OH. So please tell me the degree of alcohols. Tell me the degree of alcohols very fast. Fast. Yes everyone. This is alcohol. This is not amines. Be careful. Here, degree of carbon will decide the degree of what? Alcohol. Very good. Here, degree of carbon will decide degree of alcohols. Fine, fine. Very good. Sunil, ready? You are making some mistake. Check, please. 1, 2, 3, 4. Okay, fine. Look at this. This time, carbon will decide the degree of alcohol. Na? This carbon having OH is a primary carbon. So I will say this alcohol is also what? Primary. The carbon having OH is what? Secondary carbon. So this is a secondary alcohol. The carbon having OH is a tertiary carbon. So it's a tertiary alcohol. Right? And next one, the carbon, this one is a tertiary carbon or quaternary carbon? Carefully give me answer. Fourth one is tertiary or quaternary? This one beta tertiary na? Only one carbon, one carbon, one carbon. Tertiary, tertiary. Very good. That's why this is also tertiary alcohol. Okay, because this carbon is attached to 3 more carbons. One side the OH na? That's why it is tertiary alcohol. Okay, I hope we are clear about these things. About degree we are clear. Okay, bacha? Very good, right? Next thing is, you can see a little bit representation. Okay, some minutes we'll go to representation. Okay, of organic compounds. Clear, bacha? How we represent it? Little bit. Clear, bacha? Look at this carefully. First thing is you can say complete structural formula. Right? Second thing is known as condensed formula. Right? And which the third thing is bond line, you can say representation. Or you can say condensed representation. Fine? Bond line representation. Okay, everyone? Clear, bache? Right? So be careful. If you look at this carefully, I am writing some structure here. I have a carbon here. I have a carbon here. I have carbon and carbon, right? If I show here, I am showing its 3 hydrogens because carbon make how many bonds? 4 bonds. Here I will use hydrogen, hydrogen. Here also hydrogen, hydrogen, right? And here we have 3 hydrogens. So when you are showing some organic compound, by showing its all the bonds in a very complete way, we are opening every bond, we are showing everything. This is complete structural formula. Right? Clear? And then we see the next is condensed formula. In condensed formula, what we will write? We will write this way, like short way. CH3, this is CH3, right? Next is what? CH2. Next again what? CH2. Next again what? CH3. So here we are just showing the atoms. We are not showing their bonds, right? So here we are just showing the atoms. We are not showing the bonds. Clear, bacha? What do we call this? Condensed formula. And we want to represent in bond line representation also, right? What we do? This way. Look at this carefully. I have 4 carbons, 1 carbon, 2 carbon, 3 carbon, 4 carbon. These lines or these cuts, these points are showing us what? Carbon. So I'm showing once again, I'm saying once again, first carbon, second carbon, third carbon, 4 carbon. That's it. If in this way we are showing, it is what? Bond line representation. It is bond line representation. Fine, bache? Clear? Right? So I'm giving you one example on this. You try by yourself. Okay? Clear, bacha? Right. Very good. So please write the question. Represent in complete structural formula and condensed form, right? Or you can say representation. Okay, bache, I am giving you bond line representation. Fine. Look at this carefully. Okay. I am saying these points shows what? Carbon. So be careful. Line end shows carbon. Any joint show carbon. Any curve show carbon, right? So be careful about it, right, bacha? Next thing is, second I am saying here, we are having this thing, imagine this. This double line show pi bond. This double line will show pi bond, right? And third one I am showing here. Carefully use answer like this way. So I have given you bond line representation and you have to show the complete structure formula and the condensed one, right, bacha? Yes, everyone? Yes. This shows double bond. This shows double bond. Clear? This is pi bond here. This is pi bond. Okay, I am helping you in one case. This is a carbon. Clear? So I am showing you this is carbon. Definitely carbon will have what here? 3 hydrogens. First show short way. Carbon. What is next? Carbon. What is next? Carbon. Here also what? One more carbon, and next is also what? Carbon. Okay? Now you can show your hydrogens by yourself. Very good. The Viber is doing very well. Very good. Clear? Here also I am showing you. What is it? Carbon here first. Next carbon. This is carbon. This is carbon with pi bond. Right? Next carbon is having what? This one again, right? 1, 2, 3, 4, 5. Sorry, 5 are there, na? 5 will see. Clear, bacha? Right? Next one I am showing you again here. Carbon here, carbon here. This is carbon. Next again carbon. This carbon have 2 extra carbons. One here, one here. I can show this way. And next one again carbon, right? Every end of line or every joint is showing us what? Carbon. Clear, bacha? Clear? So now that you can add hydrogens. Now you can add what? Hydrogens, right? So please make it fast. Now at least you can give me the condensed representation. Show me, bacha. Now what we have here? It will have 3 hydrogens. Very good. Like I am showing individually. This will have 3 hydrogens, hydrogen, hydrogen, right? Hydrogen here, hydrogen. This will have 1 hydrogen. This will have how many, beta? 3 hydrogens. This will have how many? 3 hydrogens. So this is the complete representation. This is a complete representation of our bond line structure. Now if you want to go in which condensed one, so I will show this way. CH3. This is CH3. What is next one? CH2. What is next one? CH with extra CH3 with extra what? CH3. And next which one again? CH3. Okay. This way is your condensed representation. This is your condensed representation, right? Clear, bacha? Next, if you come here, this is CH here, H here, H here, right? What is it? H, H. Why? Because already it has 3 bonds. Total we have to make carbons 4 bonds. If already it is having 3 bonds, then what will come? One more hydrogen, right? It is having 2 only, so I will say H here, H here. And now what is remaining? 3 hydrogen. Right, bacha? Clear? Very good. Now if we represent in which one? Condensed one. Condensed. Okay? What do we have here? CH3. What is next thing? CH. What is next one again? CH. Or I will say again what do we have here? This is also what? This is CH2. Fine. And next one CH3. This is our condensed representation. Very good. Venkata is doing very well. Venkata is doing very well. But some students are busy in their talks. You know, out of 24-25 lakh student, only 50-60,000 get seat. Good college to very, very less. So those who will waste time will get nothing, and those who will use every single minute will get their college and will spend their entire life with respect and right. So focus on these things, bache. When you will come, these things will help you. Okay, right? So some 2-3 students, please concentrate, bacha. Next thing is this one. What we have here again? It is beta CH3. It is H here, H here. What is it? H. And here what will come? CH3. Now represent in smaller way. Represent beta in smaller way, right? Condensed one. So what I am saying here? CH3. What do we have here? CH2. This C is having beta CH3 twice here. CH3 twice, right? Because this carbon is attached with what here? CH3 and CH3, right? These two CH3 I am showing in bracket. Whenever some branching is coming, side thing is coming, we will show in bracket, right? 2 CH3 twice. Clear? Next one again what? CH3. Next one again is CH3. I hope we are clear. Right? Clear, bache? Right. In condensed form, there is no need to show pi bond. Formula is telling itself. In here you will show pi bond, right? In here you will show pi bond. In here you will show pi bond, right, bacha? I am giving one more question, then we'll move next topic. So make it clear. Okay, bache, right? Fine. Clear, bacha, right? Everyone, it's very easy, please try. I am writing the complete structure. You will write bond line and condensed, okay? Okay, this is our complete formula or complete representation with hydrogens here. Here also hydrogens, 3 will have, right? Here also hydrogens, hydrogen and hydrogen, right? Show this in both the remaining ways, like condensed one, and the next one is which one? Condensed and bond line. Condensed and bond line, right, bacha? First of all, if I say in condensed form, write this carbon having how many hydrogens? CH3. Next carbon is having 2 CH3 group, na? Extra CH3 group. So I will say C, bracket start here, CH3 twice. Clear, bacha? What is next thing? This carbon have what? CH2. This carbon is having hydrogen and show in bracket, what is it? CH3, the outer branch. And next again what? CH3, right? This is your this is your what? This is your condensed representation. Fine, bache? Now next thing if you go is a bond line. Bond line. Start here with carbon. Next again carbon. Next again this carbon. Next again this carbon. And next again this carbon. Careful. The second carbon here is having 2 extra carbon. Na? Carbon, again what? Carbon, extra carbons, right? And the second last carbon also have what? Extra carbon. This is your bond line representation. Okay? Getting my point? These two carbons are which one? This one and this one. And this carbon, beta, this extra, this one is which one? This one. Fine? I hope we are clear. Right, bacha? So now we will start IUPAC nomenclature. This is your representation, bache. Now we will start IUPAC nomenclature. Right. Okay, Venkat is saying to give one more example. Okay, I am giving last question on this. Okay, bacha. Next we will see. Next one is, I am saying here. Please look carefully. It is CH3. Here, C we have here. It is CH3. This is C. with CH3, and here we have C with H2 and CH3. Now show it in condensed form. Now show it in condensed form and show in bond line structure. Show in condensed and bond line. Right, bacha? Clear? How to show in condensed? Look at this. I can say this carbon is having two CH3 group, right? I can say CH3 twice. These are for this carbon, right? Next one. This next carbon, beta, this next carbon is having again how many extra? CH3 twice. Clear? Right? And next is what? CH2 and what? CH3. Fine? Clear? Yes, everyone. Next one is, if I say the bond line structure, what is that? This carbon, this carbon, this carbon, this carbon, and this carbon, right? Now, this second carbon have extra carbon, I am showing here. And this third carbon have extra two carbon, bacha. Here and here, right? That's why you can say it is the bond line representation. Amines, beta, amines are function groups. Amines are function groups. Amines are function group, right? Okay, very good. Yes, beta. Senpai, amines are function groups. NS2 is a function group. Okay, everyone. I hope you got the point. Fine, fine. Now, I am writing some function groups which we will use in our entire organic chemistry, right? A small point about the function groups. Okay, bache? Right. Now, next topic I am taking here, a small topic. Next is our, what is the meaning of functional groups? What is the meaning, bacha, of function group? Right? Listen to me very carefully. In organic compounds, function groups are the group of atoms which will decide all the main chemical as well as physical properties of organic compound, right? So what is that? What is function groups? I will say group of atoms, right? Group of atoms in an organic compound which decides chemical and physical properties of which decides chemical and physical properties of the organic compound of the compound, right? Clear, bacha? In a organic compound you will see a group of atoms, a group, I will say, small group, which will decide the entire physical and chemical properties of your compound. Like if I ask you, in your family, who decides all the main things in your house? Tell me, beta. If I ask you, in your family, who decides, who took the main decision of your house? You are taking or your father is taking? Tell me. In your family, who is taking the main decisions of your family? Answer me. Father. Some are saying mother also, fine. So most of the cases, okay, I'm assuming father is taking the main decision, right? So in the entire organic compound, functional group is the father in the complete family, you can say. Because functional group will decide all the decisions, all the properties mainly, right? Is bache? So you can say it is like father of the organic compound. If organic compound is containing functional group, then it will decide its physical properties and its chemical properties. I hope you are clear now. Clear? Very good. Maybe you are like just a carbon. Maybe you are just like a carbon and your father is like a function group. Clear? Okay, bacha? Very good. Fine. So we will see the examples. I am taking some examples of function group, right, bacha? First one I am saying example. We call it carboxylic group. Or wait, I will write the structure, you will tell the name. Have you seen this? COH. Tell me the name of this function group. Tell me the name of this function group, bache, fast. I know you have done in your 10th class. Can you name this function group? COH. Yes, we call this beta what? Carboxylic acid. This is known as carboxylic group, or you can say carboxylic acid. It is carboxylic acid. Very good, very good. I will write the function group, you will tell me the name. Okay, try. Next thing is, if I am saying SO3H. Have you seen this? This is known as sulfonic group. This is a what? Sulfonic group, right? Or sulfonic acid. It is which function group? Sulfonic. SO3H, sulfonic. Very good. Right. Next one, beta, I am saying third one. Maybe you will see this group somewhere. This is what this is. We call this acid anhydride. This is acid anhydride. This is acid anhydride. Fine. These function groups will come in entire organic chemistry. So please carefully. Okay. Now next thing, bacha, I am asking you. Simple. Tell me, what is OH? You know this. OH to everyone know. Tell me. OH. Yes, it's alcohol. Very good. It's alcohol. Fine, bacha. Next one I am saying NH2. Very good. What is NH2? NH2 is amine. Very good. NH2 is amine. Fine, bacha. Clear? Next I am saying, okay, we can have C double bond OH. We are having this one. Clear? But what is this? It's an aldehyde. This is aldehyde. Fine. Next one is C double bond O C. It's ketone. Next one is ketone. Right. Very good. It's ketone. Clear, bacha? Next one, if I am saying we have a CN function group. CN. It is nitrile. We call this as nitrile. This is nitrile. This is nitrile. Very good. You know about these function groups, right? You are saying right. Answering good. This is our nitrile. Clear, bache? Very good. Fine. Very good, right? Now next one I am saying we are having CONH2. CONH2 you will see. It is our amide. We call this amide. This is our amide. Clear, bacha? Very good. And next one I will say what we have here. You can say COCl. COCl. We call this acid chloride. This is acid chloride. The final one is acid chloride. Okay, bacha? Fine. Okay, done. I am saying beta once again. Look at this carefully. COH is carboxylic acid. SO3H sulfonic acid or sulfonic group, you can say. CO is what? Acid anhydride. Clear? Now, yes, CN you can say Sushant. CN you can say cyanide also, no issue. Cyanide, nitrile, both are same thing here. Okay, don't worry about it. OH beta, alcohol. NH2, amine. NH2 is what? Amine. COH beta, aldehyde. CO, ketone. CN, nitrile. CONH2, amide. COCl, acid chloride. COCl, acid chloride. Fine? Very good. Clear, clear, clear. Fine, fine. I'm asking one last question. Please answer me. Right? Tell me the number of function groups. Find number of functional groups in given question. Right? Okay, bache. Look at this carefully. We are having here CH3, CH, OH, CS2, C double bond O. Okay, C here, CN, CS2, COH. Please count the number of function groups, and you can tell me name also. Tell me the number of function groups here. Count number, beta. Yes, beta, Venkata, there are many function groups, many. We will deal with all of these in step by step, okay? Venkat, ester is there, ether is there, many are there, okay? Very good. What do we have here? First one is alcohol, OH, right? What is it? Ketone, it's keto. What is it? Cyanide, or you can say nitrile. What is it? Carboxylic group, right? Clear, bacha? So how many functions were there? 1, 2, 3 and 4. So total 4 function groups. Right. Very good. Fine. I hope you are clear. Some remaining function groups we will see tomorrow, and then we will start the nomenclature from tomorrow class. Okay, bacha? I hope this benefited you, and don't forget to fill the form in description. Okay, bacha? Bye. See you. Tomorrow we will see nomenclature and remaining some function groups. Okay, bacha? Bye. See you.
Hello, good morning students. I'm Geeta, a botany faculty. I'll be going to teach you the cell topic, cell structure and function, or the cell, the unit of life. Have you seen all your cells which are there in our body? Can you see with your naked eyes? Just we can see the lines on our body which are actually called cells. But that cell you can observe under the microscope. Are you aware of cells? I hope all of you have seen your own cells in the skin. Like I am watching my skin. There you can see the dot and lines are there. They are the cells. So these cells actually, they were discovered by Robert Hooke. You know that already, Robert Hooke. You must have had in the 10th or below your school. You have got the cell topic. So that was discovered actually by Robert Hooke. But when he discovered, it was a dead cell. It was a cork cell. That's what he discovered. But later on, other scientists was there like Anton von Leeuwenhoek. He was a person who actually discovered the living cell. So the living cells were actually discovered by Anton von Leeuwenhoek, and the first cell was discovered by Robert Hooke. But the cell which was discovered by Robert Hooke was a dead cell, and it was a cork cell. But Anton von Leeuwenhoek actually he discovered protozoan, one protozoan cell, one RBC and one bacteria. So these were actually living ones, and they were discovered by Anton von Leeuwenhoek. So the credit of discovering the living cell goes to Anton von Leeuwenhoek. But the credit of discovering the cell goes to Robert Hooke, which you already have learnt in your school time. He discovered the dead cork cell. But Anton von Leeuwenhoek, he discovered the living cells. For example, bacteria. protozoa, RBC, red blood corpuscles. So these were discovered by Anton von Leeuwenhoek. So the person who discovered the living cells was Anton von Leeuwenhoek. Then the person who discovered first time the cells is Robert Hooke. He discovered the dead cork cell of a plant. So when you are learning about the cell topic, you know, study of cells, cell structure, cells function, and all the biomolecules in the cell and all the details of cell when you are studying, that branch of biology is called as cytology. Logy means study, and study of cells. Cyto is nothing but cell. Logy means study. So study of cells is basically called as cytology. And in this cytology, you will be learning that different cells will be there. Like, you know, Anton von Leeuwenhoek discovered bacteria. This bacterium is actually coming under a prokaryotic cell, whereas these two, RBC and protozoa, they are eukaryotic cells. Eukaryotic cells, and this is the prokaryotic cell. So today's my topic is, in the cells, you have two types of cells, prokaryotic cells and eukaryotic cells. You are all aware of that prokaryotic cells. Example I have given here already, bacteria. Eukaryotic cells, we humans will come under eukaryotes. So human cells, animal cells, plant cells, fungal cells, algal cells, these all will come under eukaryotes. So eukaryotic cells means plants, animals, algae, fungi. Whereas bacterial cells will come under prokaryotic cells. So how can we differentiate the prokaryotic cells from the eukaryotic cells? There are certain differences between the prokaryotic cell and eukaryotic cells. Today, let us see only the prokaryotic cell structure. What are the features that the prokaryotic cell will be showing? All those points, let us see. I hope all of you are ready with the papers and the pens. You can take a note of all these points and you can refer to that. Okay. Today we are going to see the prokaryotic cell structure and whatever the characteristics of the prokaryotic cell are there, that ones. Okay. So now let us see the prokaryotic cell. The prokaryotic cell, the meaning of the word prokaryote. Pro means prokaryotic cell. Pro means primitive. Okay. Carry on. Wherever you get the term carry on, meaning is, this carry on meaning is nucleus. So prokaryotic cells, they contain one primitive nucleus. The oldest one, the nucleus, how it is there, that type of nucleus is called as primitive nucleus. So the cell which is showing the primitive nucleus, that is called as basically a prokaryotic cell. This primitive nucleus has another names also. This is called as nucleoid. Nucleoid. It is also called as nucleoid because there is no proper nucleus will be there in them. They don't have any definite nucleus. No definite nucleus will be there. So that type of primitive nucleus is called as nucleoid. Or one more name is there for this. This is also called as genophore. So the cells which are having a primitive nucleus or a nucleoid or a genophore, that type of cells are called prokaryotic cells. And this nucleoid or genophore or primitive nucleus, it is made up of only one chromosome. One chromosome will be present in this. You know, we have 46 chromosomes or 23 pairs of chromosomes are there in our cells, human cells. So like that, we have 46 chromosomes or 23 pairs. Here, the prokaryotic cells generally, they will be having one chromosome. Per cell, one chromosome will be there. And this chromosome will be formed forming the nucleoid, or it will be arranged in the form of a nucleoid. So the cells which are having this nucleoid are called as prokaryotic cells. Basically that is the meaning. Primitive nucleus. So the name is derived from this meaning. The primitive nucleus containing cells are prokaryotic cells. Under this you have the examples of bacteria. Examples of prokaryotic cells. They belong to bacteria, cyanobacteria. These two are the main types of prokaryotes. So these prokaryotic cells, the typical prokaryotic cell, for the explanation purpose, bacteria is a typical cell. So you will be learning the bacterial cell structure in the prokaryotic cell. Now when we take the bacteria, you already have seen that the bacteria were discovered by Anton von Leeuwenhoek. And how that bacteria will be looking. See, bacteria, they'll be having different shapes. They can be like round forms can be there, okay, or they can be rod shaped forms can be there, or they can be comma shaped ones also can be there, or they can be spring like forms, spirillum also can be there. Different forms are there. But whatever may be the shape of that bacterium, any bacteria will have a basic structure which is common for all the bacteria. The basic structure is same, but shapes may be different. And they will have generally the simple structure, but they will show complex behavior. They will be showing the simple, very simple structure, but their behavior is complex. Let us see that bacterial structure here. This is the bacterial cell anatomy. This is how one bacterial cell will be looking, and this is like rod shaped structure. This is how one bacterium is there, one bacterial cell is there, and the one which I was talking about is the nucleoid here. You can see here nucleoid is there. This nucleoid is present in the center in this way with a single chromosome. This is the nucleoid. It will be present in the center of the cell, but it is scattered in the cytoplasm. This structure is called as nucleoid. Can you see around this any nuclear membrane or nuclear envelope? It is not covered with anything. It is present in the cytoplasm only. So we can say this is naked DNA. You know already it is one chromosome and it has the naked DNA because it is not surrounded by any nuclear membrane. So the nucleoid is having naked DNA and it is having only one chromosome. One chromosome is present in the center. So this will be the nucleoid. And this DNA will be present in the center and it makes that bacterial cell prokaryotic cell because it is not surrounded by any membrane. And now the remaining part, you know, this is the cytoplasm. It will be having the cytoplasm around that nucleoid, or nucleoid is present in the cytoplasm. So you can see here, this is all cytoplasm which is opened here. This is all cytoplasm, and you can see here the cytoplasm. So inside one, around the nucleoid, everything will be the cytoplasm. And inside the cytoplasm, we have the structures like ribosomes. Small granule like things are there. They are called as ribosomes. You can see this type of granules are present. These are ribosomes. You know, in the cells generally, you must have learned that mitochondria, chloroplast, and so many other cell organelle names you must have heard. But in the prokaryotic cells or in the bacterial cells, they don't have mitochondria, ER, endoplasmic reticulum, Golgi complex. Okay, all these membrane bound cell organelles are not present in the prokaryotic cells. They don't have any other membrane bound cell organelles except the ribosomes. The only one cell organelles which are present in the prokaryotic cells are only ribosomes. Except this, all other cell organelles are absent in the prokaryotic cells. Why only ribosomes are present means because they are concerned with protein synthesis. And these ribosomes, they don't have any membrane. They are amembranous cell organelles. They are not surrounded by any membrane. So all the membrane bound cell organelles which are actually present in eukaryotic cells, they are absent here. The only one cell organelle which is present in the prokaryotic cell is ribosome. Other than this, you can't see any of the cell organelles which you know in your school, in the classes. So here the ribosomes are present in the cytoplasm, okay, and you know cytoplasm is bounded by a particular membrane. This is the membrane which is covering the cytoplasm, and that is called as plasma membrane. You can see this is the plasma membrane. So this one I am writing it as plasma membrane or cell membrane. So cytoplasm is covered by a plasma membrane. So cytoplasm is actually limited by plasma membrane or cell membrane. And that plasma membrane is always, it is living, and it will be bounding that cytoplasm, and it will be around the cytoplasm, and it will be giving one particular fine shape. After that, above that or outside the plasma membrane, you will be seeing here cell wall. So around this, one layer will be there, that is the cell wall. So this one will be the cell wall. So cell wall is actually non-living, and it will be present outer side of the plasma membrane always. So you can see cytoplasm is limited by first plasma membrane, and on outer side of the plasma membrane cell wall is present, and this is non-living. And in this picture you can see outside of the cell wall, this color, this is the capsule. So first inside one is plasma membrane, second one towards outer side we are going, outer side. First inside one is a plasma membrane, and plasma membrane outer side cell wall outer side capsule is present. This is the third one. So all these three, plasma membrane, cell wall and capsule together, all the three, they will form cell envelope. Cell envelope is formed by plasma membrane, cell wall and capsule. But the capsule is generally called as glycocalyx. So glycocalyx will be there in that. So that is, cell envelope is made up of 3 layers. The outermost one will be the capsule containing glycocalyx. First capsule will be there from outer side, then second one cell wall will be there, and inside plasma membrane. So these three will be forming the cell envelope. So it will be covering the total bacterial cell. Now outer side of this cell envelope, you can see that some projections are there with this green color, where you can see flagellae. 2 flagella, this is first flagellum and this is the second one. So if the bacterium is motile, if it is able to move from one area to another area, it can move only with the help of the flagellae. So flagella is a singular, flagellae plural. So that flagellum will be called as flagella, and they will be involved in the locomotion of this bacterium from moving from one place to another place. So this bacterium is showing 2 flagella, 1 and 2, and these flagella are the motile structures which help in the motility of the bacterial cells. Then apart from this flagellae, all of you can see in this diagram some green color hairs are there. They are called as pilae. Pilus is singular, pilae plural. So this pilae will be there, and along with that pilae some other structures also will be there. They are called fimbriae. Like this they will also be there. They are called as fimbriae. Flagella are connected with the motility. Pilae and fimbriae, they will not be concerned with motility at all, but the pilae and fimbriae helps in the attachment of this bacterial cell to the host surfaces. So pilae and fimbriae, they are not concerned with motility, but they are concerned with attachment of this bacterium to any of the host surface. So it will be attaching the bacterium to the host surface or rocks in the streams, lakes, etc. This bacterium will be attached to that particular area with the help of this pili or fimbriae. They are never concerned with the motility. So the surface structures in the bacterial cell or in the prokaryotic cell, the typical prokaryotic cell is bacterial cell. So in that, the surface structures which are present on the surface, that surface structures are three things. One, flagellum, second one, pili, third one, fimbriae. These are the surface structures which are present on the surface. So if you see here, we have covered every part which is labeled here other than plasmid. You can see here, plasmid. So the plasmid is, if you see in this diagram, it is looking like this. Okay. In this picture, if you want, this is how the plasmid will be looking. It is a single circular double-stranded DNA. It is also naked DNA because it is also not covered by any membrane. So it is a naked DNA and it is a single one, but it is double-stranded, okay, extra chromosomal. Other than chromosome it is there. This is the main chromosome, this is other than chromosome, and it is the naked DNA like the nucleoid. That is also present in some of the bacterial cells. It's not that all the bacterial cells will be having this. So some of the bacterial cells will be having the plasmids. So plasmid containing bacteria, they have an advantage over the bacteria without plasmids. This plasmid role you will be learning in the second year in the biotechnology topic. So right now the bacterial cell, which is a typical example of the prokaryotic cell, in that we are going from outside to inside. I have explained from inside to outside. Now let us go from outside to inside. So typical bacterial cell, on the surface, it will be having surface structures. Three surface structures are there: the flagella, pili and fimbriae. Flagella are concerned with motility. Pili and fimbriae, they are concerned with attachment of this bacterium to the host cells. Then below that, the surface structures, we have cell envelope. This cell envelope is having three structures from outer side to inside. The outer side, the capsule is there. This capsule is a part of glycocalyx. It is generally written as glycocalyx. Glycocalyx types are there like capsule and slime layer. So cell envelope is made up of three parts: glycocalyx, then cell wall, then plasma membrane, from outer side to inside. The glycocalyx only, it is in the form of capsule. So all the three will be having the cell envelope and they will have a particular function, protection of the cell. Each one will be having different functions. Okay, but you have to remember that cell wall is non-living and plasma membrane is living. Inside the plasma membrane, you have the cytoplasm. Total area is filled with certain matrix of heterogeneous materials. That matrix is called as cytoplasm. And that cytoplasm, in that no membrane bound cell organelles are present. No membrane bound cell organelles are present. Only one cell organelle is present, that too without any membrane. The only one cell organelle which is present is ribosome. These are the only cell organelles that are present in these cells, ribosomes. Apart from that, no other cell organelles are present. After this ribosomes, you have the nucleoid, the main part, the nucleoid. Nucleoid is nothing but, it is the primitive nucleus which is not surrounded by any nuclear membrane. No nuclear membrane, no nuclear envelope, no nucleolus. Just the single chromosome is present in the center. That too it is scattered in the cytoplasm, but generally in the center. That is called as nucleoid or genophore or primitive nucleus, I told. One chromosome. And it is generally not surrounded by any membrane, so it is called naked DNA. And it is having only one chromosome. Apart from that nucleoid, we have a single circular extra chromosomal material other than the chromosome. Certain material is there, that is also naked DNA only, that is called as plasmid. This will be giving certain advantages to the bacterium in which it is present. These are the generalized bacterial cell structures which are present in the bacterial cell structure. These are the parts, and you can categorize them into mainly the parts like cell envelope. Cell envelope will be there, okay, then surface structures are there, then nucleoid is present, then cytoplasm and other cell organelles. You know that all the cell organelles are not there except the ribosome. Now let us see that in this bacterial cell, the prokaryotic cell features, listing out the features of this prokaryotic cell. Now I have started with the characteristics. Of prokaryotic cells. The first one, they contain nucleoid or genophore or primitive nucleus. This primitive nucleus, nucleoid or genophore is not surrounded by nuclear membrane or nuclear envelope. So this is the first characteristic feature of prokaryotic cells. So they will be having the prokaryon, the meaning is primitive nucleus. And this will be made up of only one chromosome, and that one chromosome will be having one naked DNA. It will have all the genes which will control the characteristics of that particular cell. Next, they have cell envelope. Particularly bacterial cells. I have taken the example of bacterial cell and I have explained the prokaryotic cell structure. That is what given in the NCERT. So the bacterial cells will be having the cell envelope, where the cell envelope is categorized into three types. Outermost one is the glycocalyx. Outermost one. Then inside that cell wall is present. Inside that plasma membrane is present. So innermost will be the plasma membrane. So bacterial cell, it is covered by a cell envelope which is having three layers from outer to inside: glycocalyx, cell wall and plasma membrane. Inside that plasma membrane encloses cytoplasm. So cytoplasm will be there inside the plasma membrane. Then inside the cytoplasm, do they have any membrane bound cell organelles like mitochondria, chloroplast, etc.? They don't have. The only one cell organelle which is present in the bacterial cell or prokaryotic cell is the ribosome. So they don't have membrane bound cell organelles. No membrane bound cell organelles. But they have ribosomes. Because ribosomes, they don't have any membrane, so non-membranous they are. No membrane is around them, so this non-membranous cell organelles, only ribosomes are present in the prokaryotic cells or bacterial cells. Some bacteria, not all the bacteria, some bacteria, they have the plasmids. Plasmids are present in some bacteria. Which will give certain unique features for the bacterium. If they have that, they'll give the antibiotic resistance to that bacterium. So if the bacterium is having the plasmid with antibiotic resistant gene, it will not be killed by using that particular antibiotic. But it is not present in all of them. It is present in some of the bacteria. This is also naked, and nucleoid is also naked DNA. Then we have the surface structures. Surface structures are there, like flagella in motile forms. In motile forms, they have the flagella. Only motile forms will have flagella, non-motile forms, they don't have any flagella. This is the first one. Second one, pili and fimbriae. These two are not concerned with motility. Motility is not concerned with this. So the only structures which are responsible for motility in bacterial cells are flagella. Pili and fimbriae, as I told you already, they are involved in the attachment of the bacterial cells to the host surfaces. If it is motile, they will be having the flagella. If it is non-motile, the flagella are absent. Pili and fimbriae, they are present in most of the bacteria, and they are concerned with attachment of the bacterium to the host surfaces. These are all the surface structures. You have one more unique point, which is a feature of prokaryotic cell that is not there in the eukaryotic cells. Here, histone proteins are absent in the prokaryotic cells. Histone proteins means they are one particular type of protein which are not present in the prokaryotes, but they are present in the eukaryotes. And these histone proteins generally associated with the nucleus generally, but here they have the nucleoid. So in the nucleoid, these histone proteins are absent. But if you take the nucleus of eukaryotic cells, then in that the chromosomes are associated always with the histone proteins. So histone proteins are generally absent in these prokaryotic cells. It is not there, histone proteins are not at all there in any of the prokaryotic cells, and they are not associated with the nucleoid. Then prokaryotic cells, they will be storing their reserve food materials in the form of inclusion bodies. The presence of inclusion bodies is also a feature. Inclusion bodies. They store reserve food materials. In the form of inclusion bodies. So presence of inclusion bodies is unique feature of prokaryotic cells. But remember, these are also not having any membrane. They are simply present in the cytoplasm in the form of certain molecules like granules, like that, phosphate granules, cyanophysin granules, like that inclusion bodies are present. So this inclusion bodies presence is unique feature of that prokaryotic cells. And different types of inclusion bodies will be there based on the reserve food material of that organism. Whichever the food is there, based on that food, the granule will get the name. Like for example, phosphate granule, it stores phosphate. Cyanophysin granules, it will be storing cyanophysin starch. PHB granule, polyhydroxybutyrate, that substances will be stored in that. So they have got that names based on that reserve food, what is getting stored in that particular prokaryotic cell. So these are the points of the prokaryotic cells basically. You can see that list one more time. They have nucleoid, genophore or primitive nucleus. They are not surrounded by, that nucleoid is not surrounded by any nuclear membrane or envelope. Cell envelope, it is the surface structure. This is nothing but surface structure. Surface structures. Okay. On the surface it is present. Surface structures will be again flagella, pili and fimbriae. So this is on the surface. Surface structures means like you have the flagella, okay, pili and fimbriae. Surface will be covered by cell envelope, which is made up of glycocalyx, cell wall and plasma membrane from outer to inside. Inside the plasma membrane we have the cytoplasm, and after that no membrane bound cell organelles are present, and they have ribosomes. Ribosomes are present and they don't have any membrane around them. Plasmids are present in certain bacteria only, and they are also naked DNA like the nucleoid. Then they have the surface structures on the surface from cell envelope. They have the different surface structures like flagella, pili and fimbriae. Only flagella are concerned with motility. Pili and fimbriae are not concerned with motility. They help in the attachment of these bacterial cells to the host surfaces. Then histone proteins are generally associated with the nucleus of eukaryotic cells, which are absent in the prokaryotes. Because it is a nucleoid, it is not nucleus. True nucleus is not there. So the nucleoid will not have the histone proteins. And then inclusion bodies, these are the reserve food materials in these prokaryotic cells. And presence of inclusion bodies is unique feature for the prokaryotic cells. I hope all of you understood. All of you read this prokaryotic cell by tomorrow. I will be asking questions and then we will be continuing with the cell topic. Let us see the bacterial picture once here. You can see this, the bacterial structure. It is having the cell envelope, flagella, pili and fimbriae. Cell envelope is having the glycocalyx, which is capsule type here given, cell wall, plasma membrane. Then inside this is totally cytoplasm, and in this rounds are there, granule type, they are the ribosomes, the only one of cell organelles which are present here. Then in the center, the purple one is given. This is the single chromosome and it is a naked DNA because it is not enveloped by any of the membranes. Nuclear membranes are not there. So this structure is called nucleoid and it is a naked DNA which controls all the characters of that particular bacteria. Then apart from this, in the same cytoplasm, we have one more naked DNA that is called plasmid, but this is not present in all the bacteria. This is present in only some bacteria and it gives some antibiotic resistance to the bacterium which has this plasmid. So this is the basic prokaryotic cell, particularly bacterial cell structure. In this, inclusion bodies are not shown in the diagram, but I would like to show you that inclusion bodies also, they will be like this. They will be looking like granules, but they don't have any membrane. These are the inclusion bodies. I hope all of you understood that prokaryotic cell and bacterial cell. Just revise once by taking the bacterial cell diagram. You can go from outside to inside or inside to outside. Follow any one order and you have to identify all these parts. Okay. All the characteristics of the prokaryotic cells also briefly I have given you. So you have to read and learn. Thank you.
Hello everyone. Alright everyone, let's start today's class. And yesterday we have discussed immunity. So I hope you have understood immunity, right? Many like different types are also there. Yesterday we have understood only two types of immunity, that is innate and acquired immunity beta, right? But the criteria of basis of their division is also different. For example, you must have heard of active and passive immunity. Yesterday we have discussed innate and acquired immunity. Innate and acquired immunity. Yes, everyone, tell me in the comment section, tell me in the chat section, have you understood what is the meaning of innate and acquired immunity? Yes or no? Tell me in the chat section. Yes, yes. So I hope you have understood this now. Okay, excited. You all are excited. All right. So now concentrate on the class. Innate and acquired immunity, all of you have understood. Now I am going to tell you a little bit more about immunity and then I will start today's topic, that is cancer. All right? All right. So now I will tell you what is the meaning of active and passive immunity. Active and passive immunity. And then there is another type, that is antibody mediated immunity. Antibody mediated immunity. Antibody mediated immunity can also be called as beta humoral immunity. Humoral immunity. So this is one type, and the another type is cell mediated immunity. Cell mediated immunity. So these are also the types of immunity. Immunity is not only innate and acquired. There are also other types like active and passive immunity, like antibody mediated immunity and cell mediated immunity. Antibody mediated immunity is also known as humoral immunity, whereas cell mediated immunity is also known as cellular immunity. All right, cellular immunity. So now first of all, let me explain you the meaning of active. See, when you are active, for example, you are actively participating in the class. When I am asking questions, you are actively participating in the class. It's you who are doing the work. That means active. When your immune system is making the antibodies against the antigen, that is active beta. Your body, your immune system is actively participating in the generation of antibodies when there is antigen, when there is pathogen, right? We have discussed pathogens are causing the disease. So in the active immunity, the host, do you know what is the meaning of host? Host means you are host and pathogen is coming into your body. So you are the host. For example, if relatives are coming to your house, you are the host for them, right? Similarly, so when antibodies, we can write in short ABs, when antibodies are generated by host immune system, that is beta active immunity. But when ready-made antibodies are given to the patient, right? Sometimes they are naturally coming. For example, yesterday we were talking about colostrum. Yes, when a baby is born, then the first mother's milk is known as colostrum. Right? In that colostrum, which antibody is present? IgA antibody is present. So the child is receiving the antibody directly from the mother. That means the child is not making the antibody on its own. Right? So whenever the host is not making antibodies, when the host is receiving ready-made antibodies, then that is known as passive immunity. Ready-made immune response. Ready-made immune response is given. Ready-made immune response is given in case of passive immunity. For example, you can write, for example, IgA antibodies. IgA antibodies, they are present in colostrum. They are present in colostrum. What is colostrum, beta? First mother's milk, or you can say the few initial days. During the few initial days, the mother's milk will be called as colostrum. All right, first mother's milk. Or initial few days. In the initial few days, colostrum will be coming, right? Colostrum will be coming to nourish the baby. All right, everyone. So don't talk. Just focus on the topic, right? Don't talk among yourselves. Just focus on the topic. So I hope you have understood the meaning of active and passive beta. Yes, everyone. Tell me in the chat section. have you understood the meaning of active and passive? Only the meaning of active and passive. Tell me. Yes, yes. Okay. All right. All right. So now let me tell you antibody mediated immunity and cell mediated immunity. Antibody mediated immunity and cell mediated immunity. See, I have told you yesterday that we have the primary lymphoid organs, bone marrow and thymus. Yes or no? Bone marrow and thymus. From here, I have told you from the bone marrow, the B lymphocytes are getting matured, and from the thymus, the T lymphocytes are getting matured. Yes or no? So, we have understood B and T lymphocytes. We have understood B and T lymphocytes. Both of these are produced in bone marrow. B lymphocytes are matured in bone marrow, whereas T lymphocytes are matured in thymus, but both of them are produced in bone marrow. All right. Now if B lymphocyte is involved in providing the immunity, then that immunity is B cell, antibody mediated immunity. All right. And if T lymphocyte is involved, then it is known as cell mediated immunity. So the two immunities are different, beta. In one type of immunity, B lymphocyte is involved. B lymphocyte will make antibodies. B lymphocyte will produce antibodies, and these antibodies will be present in our blood, right? So humoral means what? Humoral means blood. Humoral means fluid. So here in this humoral immunity or antibody mediated immunity, this is provided by beta B cells or B lymphocytes, whereas cell mediated immunity is provided by T cells or T lymphocytes. I hope you have understood the terms. They are different terms. First type, innate and acquired. Innate and acquired is on the basis of whether it is present by birth or whether it is acquired after birth. That was the first basis, right? Second is whether it is produced by the host itself or whether it is taken from outside, right? Whether it is the ready-made response we are taking. So that was the basis. Third is on the basis of which cells are involved in providing the immunity. If B cells are involved, then it is antibody mediated immunity, because these B cells, they produce antibodies, beta. They produce what? They produce antibodies. So yes, tell me, I have concluded now the immunity, at least the types of immunity, at least you know the terms, at least you know the different terms, at least you know innate, acquired, what is the meaning, active, passive, what is the meaning, and antibody mediated immunity or humoral immunity, what is the meaning of cell mediated immunity or cellular immunity. All right, the details of all these types of immunities we will be discussing in the human health and disease chapter. All right, now in this chapter only there are two major topics. There are two major diseases. Those are, one is cancer and another is AIDS, beta. Right? You must have heard of cancer. Yes, everyone. Neela Veni. All right. Other students, please don't talk among yourselves, beta, in the chat section. Just focus on the topic. You are here to study, beta. है ना? You are not here to talk among yourselves. So just focus on the topic. And if you're not understanding anything, then just ask. Okay? Ask your doubts. You can ask your doubts and you can tell me whether you are understanding or not understanding. That's all you have to do. That is why the chat option is there for you. All right. If you will talk among yourselves, I will tell to stop the chat section. I will tell to disable the chat section. So you won't be able to interact with me. Now you see what you have to do. All right. I was telling you that two diseases are there. One is beta cancer and another is AIDS. today I will be discussing few important things about cancer, and tomorrow I will be discussing about AIDS. Alright, so let us see what is this cancer and how it occurs, right? You people can do research also in that, because that is a field where so much of the things are there which we don't know yet. Okay, we have to go into this field in more depth, and we need to know so many things which we don't know. So you can do research also in cancer, right? All right. But you are here to be the doctors, so let's focus on that only. Research you can do later on if you have other plans, right? All right, everyone, now starting the cancer. All right, starting the cancer. So beta, cancer is, do you know that it is the major cause of death? Do you know annually 1 million Indians suffer from cancer annually? In every year, 1 million Indians suffer from cancer, and majority of them die from cancer. All right. So cancer is the major cause of death. If somebody will ask you directly this question and give you 4 options, which of the following is the major cause of death? Answer will be cancer. It is the exact line of NCERT beta. All right. So cancer is the major cause of death. It is the major cause of death. Now, first of all, what is the meaning of cancer and what happens in cancer? Beta, in case of, when, do you know that in your body, there are some mechanisms, like for example, cells are dividing, okay? Cells are dividing, cells are growing. All these mechanisms are highly regulated. Yes, beta. Yes, cigarette is also a cause of cancer, Swati. I will tell you what are the causes of cancer. I will tell you. But first, let us discuss, Ganesh, the meaning of cancer. Yes, exactly. Ma'am, just like cancer loves death, we love your class. Oh my God, thank you. Thank you, my lovely students. All right, now let's focus on the topic, cancer, meaning of cancer. So beta, when these cells of your body, when they are, dividing and growing in an uncontrolled manner. See, sometimes we can control things, right? But sometimes something will happen, due to that reason, the things are going uncontrolled. So when the cell division, when the cell growth is going uncontrolled growth and division of cells, then so many cells will be, mass of cells will be formed, and that is known as tumor. So let us write, cancer means uncontrolled growth and division of cells. Uncontrolled growth and division of cells. It will lead to what? Uncontrolled growth and division of cells, leading to formation of mass of cells, and this mass of cells is called as tumor. Tumor beta, tumor. Or it can also be called as neoplasm. So uncontrolled growth and division of cells leads to the formation of what? Leads to the formation of tumor. You must have heard of tumor. So now you understood what is the meaning of tumor. Tumor is the mass of cells beta, mass of cells, group of cells. So many cells are there. How these cells have formed? These cells have formed because of the uncontrolled growth and division. They are continuously dividing. All right. Yes, Ganesh, there are medicines for cancer, beta. I will tell you the therapies also. All right. I will tell you the therapies also. But first, step by step, we will go into the topic, step by step. So, you understood how a tumor is formed. So, now you understood in a cancer, tumor is there. Beta, Doc Lucky, is there any difference between cancer and tumor? Beta, in case of cancer, cancer is the name of the disease. In case of cancer, tumor is getting formed in the body. Tumor is getting formed in the body. Okay, have you understood now? So now tumor. The cells of this tumor can be called as the cells of this neoplasm or tumor can be called as cells are known as neoplastic cells. Neoplastic cells. Now don't get confused. They are not made up of plastic, beta. They are not made up of plastic. Don't get confused. These are simply known as neoplastic cells because they are newly formed cells. Neo means new. They are newly formed cells, right? They are neoplastic cells. Now, beta, there are two types. There are two types of cancer or this tumor. We can call two types of tumor. Two types of tumor can be formed. Two types of tumor can be formed, right? One is beta benign tumor. Benign, B-E-N-I-G-N, benign tumor. Benign tumor. And another is beta malignant tumor. Malignant tumor. Malignant tumor. Alright. Benign tumor is a tumor which is, occurring at one site, at one place, any place in the body, it is occurring, right? Then it does not go to any other place. It is occurring at one place and it will remain there only. Okay, it will remain there only. It will not go anywhere else. So that is the benign tumor. So can we write benign tumor is a localized tumor? Localized means it remain confined. It remain confined to the site of its origin. To the site of its origin. Where it has formed, it will stay there only. Where it has formed in the body, it will stay there only. It will not go anywhere else. But this malignant tumor is very, very dangerous. Because in this malignant tumor, there is a property of migration. In this malignant tumor, if tumor is formed at one place, it will migrate to other places and then again it will cause tumor at various places in the body, right? So malignant tumor is not localized. It can migrate. It can migrate to different sites. It can migrate to different sites. Right? How it can migrate to different sites? It can migrate, yes beta, you can always take the running notes, beta Nikhil. You have to, notes, you should make notes. Whenever you are studying anything, whenever you are even simply reading also a book, then also you should scribble on the side. You should always write because writing will help to remember. If you won't write, if you only listen, you won't remember. You will forget easily. Okay, so writing is important, writing is necessary. I will tell you all these tips and tricks also to remember, right? Once you come here. But since we have less time, we cannot talk much about all these things. So let us focus on the topic, right? So beta can migrate to different sites. How? I was telling you how it migrates. So it can migrate through the blood because blood is going everywhere, right? It can migrate through the blood. Also, it can invade the neighboring tissues, neighboring cells, it can invade, and through those neighboring cells, it can go to different places. So this property of migration is known as metastasis. So this property of migration is known as metastasis. So if somebody will ask you, metastasis is shown by which tumor? Benign tumor or malignant tumor? Benign tumor or malignant tumor? So now malignant tumor has this metastasis. Very, very important property. Ma'am, if cancer came to some sensitive part, okay, beta treatment is there. Cancer can be, cured, but obviously not all the cancers, but yes. And if cancer is diagnosed at early stage, then the cure is possible, then the treatment is possible. But obviously when the cancer has spreaded so much, then it is not, right? Then it is not. All right, malignant. So malignant cancer has the property of metastasis. Malignant tumor has the property of migration. That property is known as metastasis, right? Now I will tell you one more property. Property of cancer cells, property of tumor or maybe cancer cells. One more important property is the formation of new blood vessels. See, whenever in your body new cells are forming, they require nutrition, they require oxygen, they require glucose, they require food, है ना? Everything they need, right? So then there is a process takes place that is known as beta angiogenesis. Angiogenesis means what? Angiogenesis means beta formation of new blood vessels. Formation of new blood vessels. Formation of new blood vessels. New blood vessels, right? Why angiogenesis is required in case of tumor? Because in tumor many cells are formed, and all these cells which are newly formed, they require nutrition, they require oxygen. That is why blood vessels will be formed, and then blood will be taking these nutrition, growth factors, oxygen to the newly formed cells. It will be supplying. So these blood vessels, new blood vessels, formation of new blood vessels for the supply of nutrients, oxygen, growth factors, etc. Because all these things are required by the nutrients, oxygen and growth factors. All these things are required by the newly formed cells. All right. So whenever there is a tumor formation, be it benign tumor, be it malignant tumor, angiogenesis occurs. We will talk about chemotherapy, beta. Just wait. So angiogenesis occurs in the case of tumor. Whenever there is tumor formation in the body, then angiogenesis takes place, right? Now let me tell you one more thing. See, if you are a good person, tell me everyone, if you are a good person, then you will be having so many friends, yes or no? Yes or no? You will be having so many friends if you are a good person, है ना? But if you are a bad person, then nobody wants to stay in contact with you. Nobody would like to stay with you. Yes or no? Yes or no, everyone? Yes? Are you understanding? I will explain you something which is very, very important related to cancer. So just listen to me carefully. If you are a good person, you will be having so many friends. These are your friends. If you are a good person, these are your friends. If you are a good person, these are your friends. Yes or no? Yes or no? But if you are a bad person, nobody will make contact with you. See, they are far. They are not making contact with you. Yes or no? Are you understanding this? Right? Just like that. Good person is a normal cell, beta. Good person is a normal cell. Good person is just like a normal cell. And bad person is just like a cancerous cell. Bad person is just like a cancerous cell. So normal cell is having a property. Normal cell has a property. Normal cell shows the property of contact inhibition. The name of the property is contact inhibition. Sorry. So, contact inhibition. See, contact means what? Contact, Shravani. All right, Malika Arjuna, your name is Shravani. All right, beta, I will try to remember. All right. So now, good person is just like a normal cell. So, normal cell has contact with other cells. Yes or no? So, normal cell has the property of contact inhibition. Why the contact? When you have contact with others, it will help you. For example, when you are a good person, when you are a good student, I will try to help you. Na? If let us suppose you are doing something wrong, if you are making mistakes, I will stop you to do those mistakes. Yes or no, everyone? I will definitely stop you to do those mistakes. If you are a good person, then your friends will stop you to do something which is wrong. Yes or no? Yes or no? If you are a good person, your friends which are in your contact, they will stop you to do something which is wrong. That is why normal cell is not showing uncontrolled growth and division. Why? Because the cells which are in contact, they will stop this. They will stop the uncontrolled growth and division. So, contact inhibition is a property by which the adjacent cells stop, or you can say regulate the uncontrolled growth and division, of a normal cell, But see, read it once more. Normal cell shows the property of contact inhibition. By this property, adjacent cells, these are the adjacent cells, beta. These are the adjacent cells. They will stop the uncontrolled growth and division of the normal cell. Are you understanding? Are you understanding? Are you understanding, everyone? I am not strict. If you will not listen to me, if you will not listen to my class, and if you will not follow my instructions, then I am strict. But if you are doing everything what I am telling you to do, then I am very cool. Alright. So now, beta, angiogenesis is the property of the tumor. Rian, angiogenesis is the property of tumor, beta. Wherever tumor is formed, angiogenesis, you will see the angiogenesis also. So all of you have understood this. Normal cell. Now cancerous cell. Cancerous cell does not have this property. Cancerous cell shows loss of contact inhibition. What is the property of cancerous cell? Loss of contact inhibition. Once you were a good person, then due to some reason you became bad person. When you became bad person, then your friends also detached with you. Then your friends stopped talking to you. Alright. Similarly, now this is a cancerous cell. It has lost the contact inhibition. That is why we say cancerous cell has the property of loss of contact inhibition. So if somebody will ask you, contact inhibition is the property of. Contact inhibition is the property of. Contact inhibition is the property of normal cell or cancerous cell? Contact inhibition is the property of normal cell or cancerous cell? Yes, everyone, give me the answer. Give me the answer. No, Rohit, Dipti, no. Normal cell. Contact inhibition is the property of normal cell. Dipti, beta, Hussain, contact inhibition is the property of normal cell. But when somebody will ask you, Soumya, not the cancerous cell, not the Shravan, not the cancerous cell, beta. Some of you are giving me wrong answer. Contact inhibition is the property of normal cell. Loss of contact inhibition is the property of cancerous cell. Loss of contact inhibition is the property of cancerous cell. That means here in this case, adjacent cells will not stop. Adjacent cells will not stop, will not regulate or stop the uncontrolled growth and division. The uncontrolled growth and division. It will not stop, and that is why cancerous cell will keep on growing and keep on dividing, and that's how the cancerous cell will make the tumor. So yes, everyone, now tell me, have you understood the difference between Krishna, have you understood the difference between the contact inhibition and loss of contact inhibition? When you are a good person, your friends are in contact with you and they will stop you to do something wrong. But when you are a bad person, nobody will stop you, nobody will come near you, nobody will tell you that you are doing wrong, and you will keep on doing wrong. That's how normal cell have contact with other cells, have contact with adjacent cells. These adjacent cells, they will stop the normal cell to grow or divide in an uncontrolled manner. But if there is a cancerous cell, it has lost this property. Cancerous cell, if there is a cancerous cell, the adjacent cells will not stop it for the uncontrolled growth and division. Alright? Have you understood? Everyone tell me in the chat section. Have you understood normal cell has contact inhibition? Cancerous cell, no contact inhibition. Loss of contact inhibition. Very, very important property, and it can be asked in the NEET exam. It can be asked in the questions. Now I will tell about the cause of cancer. How this cancer is occurring in the body. Okay, cause of cancer. So what is the cause of cancer? Let us talk about the cause of cancer, beta. Cause of cancer. So now I will tell you something, that in a cancer cell, beta, see, first of all, let us start with a normal cell, because once the normal cell will be a normal, once a cell will be normal, then something will happen due to which it will turn into a cancerous cell. Alright. So what will happen? Normal cell has genes. Those genes are known as cellular oncogenes. Or they can be called as proto-oncogenes, beta. They can be called as proto-oncogenes. So every cell of our body has cellular oncogenes or proto-oncogenes. Beta, cellular oncogenes or proto-oncogenes, they are inactive genes. They are, beta, inactive. They are not active. But if they get activated, then what will happen? If these cellular oncogenes or proto-oncogenes will become oncogenes, that means this is active. That means what? That means this is active. That means this is what? This is active, beta. So then this cell will be called as a cancerous cell. So how this oncogenic transformation is occurring? Transformation means change. The cellular oncogene is converting into oncogene. How this transformation is occurring? This transformation is occurring due to the beta carcinogens. Carcinogens are cancer causing agents. So these are carcinogens. What are these carcinogens? Cancer causing agents. these cancer causing agents, they will do this oncogenic transformation. Oncogenic transformation. They will convert the inactive gene into the active gene. So yes everyone, what is happening? How a normal cell is converting into a cancerous cell? Yes everyone. Neela Veni, I remember beta. I remember all the names. All right. Now you tell me, everyone tell me, did you understood how a normal cell is getting converted into a cancer cell? Normal cell has the inactive genes. When these genes are activated by carcinogens, then the normal cell will become cancerous cell. Yes or no everyone? All right. So now let us talk about the carcinogens. Now let us talk about these carcinogens. So there are different types of carcinogens beta. Carcinogens are cancer causing agents. They can be beta physical carcinogens. There can be chemical carcinogens, and there can be biological carcinogens. So examples of physical carcinogens are beta radiations. Do you know radiations can cause cancer? Do you know that? Yes everyone, tell me in the chat section. Do you know that radiations can cause cancer? The sun radiations, the radiations coming from the sun, that is why you should not come into the exposure of sunlight very much, very much. Yes, that's why we apply sunscreen. Yes or no? To just protect our skin from the harmful radiations of sun, we apply sunscreen. All right. So radiations. What are these radiations? There can be ionizing radiations. For example, X-rays, gamma rays. X-rays and gamma rays. X-rays and gamma rays. There are non-ionizing radiations also. For example, ultraviolet radiations, or you can call them UV rays. All these radiations can cause cancer beta. All these radiations are carcinogens. They can do this oncogenic transformation. They can convert a normal cell into the cancerous cell. Thank you, Sankar Manna. Thank you, beta. All right. Now, chemical carcinogens, beta. Now, somebody was talking about cigarette. Yes or no? That cigarette smoke or the tobacco smoke can also do the cancer, right? Can also be a cause of cancer. So many carcinogens are there which are chemical carcinogens. We are using so many things in our food item. We are using so many things in the cosmetics, and these chemicals can be carcinogen. So any chemical which can convert normal cell into cancer cell, that will be chemical carcinogen. For example, you can write tobacco smoke. Tobacco smoke is a kind of cancer. Soot, coal tar, mustard gas, aflatoxin, diethylstilbestrol. Beta, there are many chemicals which are carcinogens. I'm not writing all of these because only tobacco smoke is written in NCERT. But I'm telling many chemicals are there, right? So all these chemicals are chemical carcinogen. They will cause lung cancer. Tobacco chewing will cause oral cavity cancer beta. So many types of cancers will be caused by the chemicals. Biological carcinogens are beta viruses. There are some of the viruses which will lead to the formation of cancer. For example, HIV. HIV is causing AIDS, but it is associated with one type of cancer also. So there can be so many viruses. For example, human immunodeficiency virus. For example, human papilloma virus. For example, herpes simplex virus. Many viruses are there. Beta. Okay, they can be called as biological carcinogens. So now you understood the cause of cancer, right? You understood how cancer is occurring. Yes, everyone, tell me quickly in the chat section, have you understood this? Then I'm going to quickly tell you two things. One is how we can detect cancer, and the other thing is how we can treat cancer. Only two things are left. Only two things are left. One is detection and other one is treatment. So let me talk about the detection first, and then I will talk about the treatment. Beta, detection. So many techniques are there. Detection, and I will talk about the treatment, beta, treatment, right? There can be different types of cancer, and for different cancer, no beta, TB is not a type of cancer. TB is not a type of cancer, beta, Ritvik. All right. So now, talking about the detection or diagnosis or detection, for different cancer, it can be different. All right. We have different approaches, beta. For example, if I talk about blood cancer, WBC, we can detect blood cancer even by a simple blood test or by a bone marrow test, right? So there are different detection techniques. Let me tell you those which are written in NCERT. Beta, one is surgery. That is the treatment. One is beta X-rays. We can use X-rays. Then we can use CT scan. Then we can use MRI. You must have heard of all these techniques, beta. Yes or no? You must have heard of all these techniques. X-rays, CT scan, MRI. Then there is one more technique that is known as antibodies. Antibodies. Yes, you will be amazed to know that we can also use antibodies to detect cancer, beta. Okay, so we can use X-rays. Even in the CT scan also X-rays are used. X-rays are used in the CT scan. X-rays are used. But in case of MRI, magnetic resonance imaging, CT scan is computed tomography scan, which uses X-rays. MRI uses strong magnetic field. It uses strong magnetic field, and also non-ionizing radiations. Non-ionizing radiations. Both of these techniques, CT scan and MRI, both of these techniques, they are giving you the 3D imaging, beta, of the internal organs. 3D imaging of the internal organs, 3D imaging, right? But if somebody will ask you the question that which of these technique is the safest technique to detect cancer, that is MRI, beta. MRI is considered as the safest technique to detect cancer. Why? Because it is using magnetic field. It is using more magnetic field, not more of the radiations. You know, radiations can be harmful. X-rays are harmful, beta. They are harmful. Harmful, right? Harmful radiations. So, but we can use X-rays to detect cancer if only for a very less period of time. If we'll give the X-rays, then they won't do the harm, right? They won't be able to do the harm. So you can remember X-rays, CT scan, MRI, antibodies. We will talk about how antibodies can detect cancer. We will design these antibodies. We will make such kind of antibodies which will detect the cancer cells, right, in the body. They can detect the cancer cells. Now talking about the treatment, you must have heard of surgery. Surgery is done, right? First of all, before surgery, histopathological studies are done, beta. Histopathological in the detection, not in the treatment, beta, in the detection. Histopathological studies. What is the meaning of histopathological study? Suspected tissue in the body. Suspected tissue is cut and examined. Suspected tissue is cut and examined under microscope, I will take 2 more minutes and then I will conclude the topic. So don't go anywhere, listen to me. Alright, those who are understanding the topic, just listen to me. Histopathological studies are beta for the detection. If let us suppose we are feeling that there is some suspected tissue where tumor is there or a kind of cyst is there, right? So we will cut a part of that tissue and we will examine under the microscope to see whether the cells are dividing in an uncontrolled manner, so that we will tell that okay, this is a tumor. Once we will get to know that this is a tumor, we will do the surgery. So after histopathological studies, surgery can be performed. The whole tumor will be removed by surgery. The whole tumor will be removed. Okay. So this is surgery. But sometimes in case of malignant tumor, we cannot perform surgery at many sites. Right. So at the major site, we will perform the surgery. Along with the surgery, we will use other treatments also. Like along with the surgery, we will do chemotherapy, chemicals, medicines will be given, right? Then we can also do radiotherapy, radiations can be given, and we can also do the immunotherapy, in which we will not give antibodies. Immunotherapy, we will give alpha interferons beta, biological response modifiers. Surgery, you understood. Chemotherapy, chemotherapy is beta chemicals, medicines, vinblastine, vincristin. These are some type of medicines which are given. They are extracted from plants only. Radiotherapy, radiations, harmful radiations to kill cancerous cells, harmful radiations to kill cancerous cells, right? But obviously there are side effects beta. Chemotherapy side effects are there, hair loss, anemia. Radiotherapy also side effect is there. Normal cell will also be dying, okay? Because of the harmful radiations, normal cell will also be dying. Immunotherapy, in immunotherapy what we are giving? We are giving biological response modifiers. Biological response modifiers. What are these? These are nothing but alpha interferons. They are given so that our immune system will also fight with the cancerous cell, so that our immune system will be supported, and then it will fight with the cancerous cells. All right. So this was all about cancer. I have tried to tell you everything which is important in case of cancer. Now if you want, you can read it from NCERT and then you can come back and tell me if you have not understood anything. Right? Tomorrow we will be discussing AIDS. So see you tomorrow. Like the session, subscribe the channel. Bye bye. Take care. God bless you all. See you tomorrow.